All right, everybody. So what we're going to talk about now is some of the stuff in that horrible supplemental reading that I made you guys read. I know it's complicated. I'm joking when I say it was horrible. But what I want to do is, I know it's complicated. I'm hoping that reading elucidates some of this epistatic stuff a little bit. And I wanted to give it to you guys specifically because there's two very important genetic techniques that are not talked about in your text. That are talked about in supplemental reading. So what specifically is in that supplemental reading? Well, there are two specific descriptions of techniques: genetic dissection and complementation testing. And these are ways that geneticists can study how genes interact with each other. And we'll start with the description of genetic dissection and then go on to complementation testing. So this kind of harks back to the purple flowers that we looked at in the previous lecture. And remember that it's normally true that more than one gene contributes to a single phenotype. This is also called a trait like this, which is almost every trait, is called a polygenic trait. Polygenic meaning more than one gene or multiple genes. And that's because oftentimes to produce a phenotype or a trait, you need to do something complicated in the cell. And oftentimes there's some kind of genetic pathway that is required to produce a phenotype. And what genetic dissection allows you to do is figure out where a gene acts in the genetic pathway. So let me explain what I mean by that. Genetic pathways are essentially sequential steps. Each step is governed by a single gene, and you've got to do all the steps in a row before you get to the end and make something that gives you a phenotype, basically. There are three main types of genetic pathways that are studied. We're just going to concentrate on this first one, a biosynthetic pathway. It's the simplest of the three. So here's an example of biosynthetic pathway. This is, of course, a made-up one. And this is similar to getting the purple pigment that we saw in the sweet pea exercise previously. So what we want to make here is compound D. A, B, C, and D are all separate chemicals for compounds. When you look, and so the idea, right, is that in order to get from A through to D, D being something we want as an organism, we have to take A chemical and we have to modify it a little bit until it becomes B. That's a chemical reaction. B has to be modified in some way and become C. And finally, when we get C, we can modify C to get what we really want, which is D. So this describes 1, 2, 3 chemical reactions that are occurring, right? Each one of these chemical reactions is facilitated by an enzyme. So enzyme number 1 catalyzes the conversion of A to B. B would be an intermediate in this process that needs to be catalyzed by enzyme 2 to become C, another intermediate. And then C needs to be catalyzed by enzyme 3 to become D. This C would be another intermediate. And remember that these enzymes are all just proteins, and they're encoded for by genes, right? If you mutate the gene that makes enzyme 1, it can no longer do this conversion because it cannot make the protein enzyme 1. Now, there's a couple of really important assumptions when you're looking at biosynthetic pathways and thinking about genetic dissection. One is that there are sequential steps here in a linear order. You have to do everything in order. If you start with A, the only way you can get to D is by going through B and C to get to D. You can't take A, skip B, and go to C. Right? You've got to do it in order. It's important to remember that when you're looking at dissection. So this was originally done by Beadle and Tatum, these two scientists. They were actually looking at bread mold, because, why not? And they were looking at requirements for bread mold to grow, basically. And they looked at some biosynthetic pathways in this organism. So what they did was they took a bunch of bread mold and they mutated it and asked what those mutated bread molds needed to grow. And what they showed was that single mutants could be responsible for different steps in a biosynthetic pathway. And that led to this hypothesis known as the one gene, one enzyme hypothesis. It's a very famous hypothesis in biology. So here's what they did. This is a tube of growing mold cells. These mold cells are growing on what is called minimal medium. What does that mean? That means, so these bread mold cells, they need all of the amino acids that, the 20 amino acids that we use in biology. They need all of those to grow. If they don't have one of those amino acids, they're dead. This minimal medium does not contain any of those amino acids. What it does contain, though, is precursors that this mold can use to make those amino acids. So this is wild type bread mold, growing quite happily on this minimal medium because it has lots of enzymes and it can make its own amino acids. What we're going to do is zap this thing with X-rays. So right now, we know that these must be wild type functional cells because they're growing on minimal medium. But what we're going to do is zap them with some X-rays, and then we're going to take single cells and we're going to transfer them to complete medium, which contains all the amino acids they need. So even if they can't make their own in step 2 here, they'll be fine because we've given them all the amino acids they need. And each one of these tubes is meant to show you a single mutant. So the fact there's a bunch of red spots on here just means it started off as one cell and then it grew up to be nice and healthy. So each one of these tubes represents a single mutant in a single gene. You don't know what genes they are because it's random, the way the X-rays mutate DNA. But you know that in each one of these tubes is a single mutant. And then what you're going to do is you're going to take each one of these tubes and you're going to transfer some of these cells from complete media, where all the amino acids have been added, back to minimal media, where there are no amino acids. You got to make your own. And what you see here is that most of these cells, most of these mutants grow just fine, but here's one that doesn't. So that means this guy, when you took it off complete media and put it onto minimal media, it can't grow. And that means it must have a mutation in some pathway that makes some amino acid because it dies on minimal medium. You could actually tell that it's amino acids that it's missing because you can do this kind of cool experiment where you take this one mutant, it won't grow on minimal media. You add all the amino acids on minimal media, it grows. If you add vitamins to minimal media, it can't grow. If you add nucleic acids to minimal media, it can't grow. But if you put it on complete media, it can grow. So these tubes, these results suggest that this mutant won't grow on minimal media because it's lacking one of the amino acids, because it can't make one of those amino acids. Of course, we could add each individual amino acid to the media and see which one it requires for growth. And as you can see from this experiment down here, this particular mutant requires methionine for growth. Because if you add methionine, which is just one of the amino acids, it grows. Which suggests that this thing dies on minimal media because it can't make its own methionine. So what we've done is we have created a mutant that can't make methionine. Great. The next question they had was, why can't it make methionine? And this is where we look at the real genetic dissection of this pathway. So this pathway that you're looking at, this biosynthetic pathway, is an actual biosynthetic pathway, similar to the one I showed earlier, which just said A to B to C to D. In this case, we're showing you real chemicals, real intermediates. So methionine over here is what we need to make to live. That's an amino acid. In order to make it, we start with a chemical called homoserine. There is an enzyme MET4, which again, there's a gene that makes this protein. MET4 is required to catalyze the conversion of homoserine into cysteine. If we make cysteine, MET3 is another gene different from MET4. MET3 makes an enzyme that converts cysteine to cystathionine. MET2 is an enzyme that converts cystathionine to homocysteine. And finally, MET1 is an enzyme that converts homocysteine to methionine. And what Beadle and Tatum wanted to discover was, similar to the experiment I just discussed, where we're adding back amino acids to figure out which one is required, if we could add back these intermediates, maybe we could figure out where the pathway is blocked. And Beadle and Tatum knew this biosynthetic pathway because this was already quite well established. And when they did this experiment to look for mutants that couldn't make methionine, they didn't just find one, they found a whole bunch of them. And so they were wondering, could some of those mutants be for this gene? Could some be for this gene, some for this gene, and some for this gene? And they did an experiment to find that out. So here are 4 different mutant strains that they found. MET1, MET2, MET3, MET4. MET1, MET2, MET3, MET4. These mutants are all mutant for the production of different. And then they knew that these guys did not grow on minimal media, because that's how they identify them in the first place, right? They know they don't grow on minimal media. They know when they add methionine, all of these mutants should grow because that's how they found them in the first place. So if we grow these things on minimal media, so these are the mutants down here, and across the top is what they're trying to grow these guys in. Minimal medium alone, none of them grow. This is a positive control of a wild type strain. And then if they added methionine to the minimal media, they would all grow. And we knew that was true because that's how they were isolated. So, and that makes sense, right? Because methionine is at the end of this biosynthetic pathway. So what they wanted to do was, for example, they wanted to take a MET3 mutant and add back different intermediates and see when it would grow and when it didn't grow. And what you see is that when you add methionine, a MET3 mutant can grow. That makes sense because that's giving it what it needs, not asking it to make anything. If you give a MET3 mutant homocysteine, it can grow just fine because it's a MET3 mutant. It can't carry out this step, but a MET3 mutant is wild type for MET2 and it's wild type for MET1. So if you give it homocysteine, it can convert homocysteine to methionine because it has this enzyme, not a mutant for that. If you give it cystathione, as shown here, you get a little plus because it grows, right? Because it has a normal MET2 and a normal MET1, so it can do this and it can do that. But if you give a MET3 mutant cysteine, as shown here, you get a minus, which indicates that it didn't grow. And that's because a MET3 mutant can't do this conversion from cysteine to cystathionine. hoping that makes sense. And you can look at that, and you can actually figure out where MET3 is in this pathway, because essentially, if you add the intermediate after the pathway is blocked, it grows. And if you add the intermediate after the pathway is blocked, it doesn't grow. MET1, for example, doesn't grow on any of these media where you add any of the intermediates. It only grows on methionine, which means it must act here in the pathway, because if you add this, it can't grow, and if you add this, it can't grow, and if you add this, it can't grow. So if you add cysteine, if you give a MET1 mutant cysteine, it contains a functional enzyme for this conversion, so it makes this. It contains a functional enzyme MET2, so it makes homocysteine, then it can't make methionine. So the only thing you can add to make a MET1 mutant grow is methionine. And that is how you genetically dissect a pathway. You're sort of taking a mutant, you're adding minimal media, you're adding different things to minimal media, seeing what it can grow on and what it can't. You can use that to order the pathway, where the genes are. Okay, the one other technique I want to talk about is complementation testing, which is essentially a simple way to ask to answer this question. So if I isolate two mutant alleles that have the same phenotype, there's a possibility that I just basically made two different mutants in the same gene. Or I could have made mutants in two different genes that are involved in the same phenotype. And in order to figure that out, you can do what is called a complementation test. And so the way that you do this is very similar to an experiment you already know about. So if you remember back to that experiment in the sweet pea, where we took two white flowers and we crossed them together, the progeny were purple, and that was an example of a complementation test. So we had two mutants that were both white. We brought those mutations together into the same plant by crossing the two white flowers together. And we saw a purple flower result from that cross, which is the normal or wild type phenotype for the pea plant. And so what that showed was the two mutations complemented each other, showing they were in two different genes. So here's another way to envision that. Here's an experiment done in fruit flies. Fruit flies normally have red eyes. That's the wild type phenotype for fruit fly eyes. But to get a red eye, you need to go through a biosynthetic pathway that has multiple steps. So again, a white eye can be converted to a red eye by doing a couple of different reactions which are going to be catalyzed by different enzymes. So here we have an example of two white-eyed fly strains. They're white for different reasons. This white eye is because it's homozygous for gene B, which is required at the second step in this pathway. Over here we have a second strain, which is also white-eyed, same phenotype, but for a different reason. This is homozygous mutant for gene A, which is required for this intermediate step in this pathway to red eye. So if we take two white-eyed flies that are both homozygous mutant for B, and we cross them together, or A, it doesn't matter. So, okay, here we have two white-eyed flies that are like strain 2. They're homozygous for the non-functional A allele. Cross these together, no rescue takes place because you have homozygous little a, homozygous little a. There's only one thing that the progeny can be. This is a true breeding strain. It stays white. But if I cross this guy, strain 1, with this guy, strain 2, and we make a a fly here. This fly is going to have red eyes because the wild type version of A from this strain has rescued the mutant A from this strain, giving us a functional first step in this path. On the other hand, the capital B from this strain of flies rescues the homozygous recessive B from the first strain. Therefore, we have a functional step B, and we get a hybrid that has red eyes. This is called complementation, where we get one wild type from each strain, rescuing the mutant from the other strain. This is an important concept to understand, and it's actually important because you can use it as a tool. So imagine that I was a Drosophila biologist, which for me is not much of a stretch because I was for many years. If I was mutating flies and I had 9 different mutants, for example, that all had white eyes, I would like to ask the question, well, you know, are these 9 different alleles of the same gene, or are different genes involved in all of these different mutations? I could find that out very simply by simply crossing each of the 9 strains to every other strain. And if there's a complementation, in other words, if the flies that result from that cross have red eyes, I know that the 2 white-eyed flies have mutations in different genes, right? Because it's just like this situation. If I cross 2 white-eyed flies together and they don't complement, in other words, if this situation occurs, it must mean that the 2 flies are mutant in the same gene because there was no complement. And that allows me to sort of, instead of studying 9 different mutants, I can cut that number down, because if 5 of these are in the same gene, then, you know, I just want to study that gene. So this is called a complementation matrix, and it looks like this. So here we have exactly the experiment I was talking about. We have 9 different mutations from all different names: apricot, brown, buff, carnation, cherry, claret, coral, vermilion, and white. These are all different white-eyed fly mutants, 9 of them. And what I'm going to do is I'm going to cross them all together with each other and see if the phenotype is rescued. So if I cross apricot with apricot, if I do a self-cross, there is no rescue, which means I put a minus on my complementation matrix. Which makes sense, right? I know that these 2 mutants are the same gene because I isolated them, they're the same thing. So if I do a self-cross, I'm always going to get a minus, because that's just like this situation, right? I just basically crossed the same thing together. No complementation occurs. And if you look down the complementation matrix, you'll see that whenever you cross something to itself, you always get a minus. Now, if I cross this apricot mutant with a brown one, so this is the cross. So here, apricot is crossed with brown. Check it out. I get a plus, which means these guys have a rescued phenotype. They have red eyes. So I know that this mutant and this mutant must be in different genes because I've got a complementation effect, just like you see. And by crossing everything with everything else, I can essentially ask how many of these mutations are in the same gene. And if you look across this top row, you'll see that if I cross apricot to buff, no complementation. Apricot crossed to cherry, no complementation. Same with apricot crossed with coral and with white, which means that the mutants white, coral, cherry, buff, and apricot are all in the same gene. And that's great because it allows me to put all those mutations in one box and say, hey, these are all in the same gene. Whereas apricot and carnation must be different genes. So on and so on and so on. If you look at this table and you interpret it using these minuses, you'll see that we have what are called different complementation groups. And that just means that if anything doesn't complement anything else, you put them in the same complementation group. So apricot is basically doesn't complement buff. You put those in the same group. Cherry, coral, and with white. And I know that all of these mutants are in the same gene. That's why I put the same complementation group. And when I do that with this entire complementation matrix, what I see is that we're actually studying 5 different genes here. We've got 1, 2, 3, 4, 5 mutations in gene 1, and then everything else, we have 1 mutation in these genes because there's no other complementation that occurs in this matrix. So that's how complementation matrices work. And there are some problems that you're going to do revolving around those in your little packet. Good luck.